Welcome back to the lecture series in animal physiology. So, we concluded the last lecture with a very brief introduction about spatial and temporal summation in the neural networks. So, we are into the 6th week and this is the first class of the 6th week. So, here we will elaborate this further, what we exactly kind of physiological significance of spatial and temporal summation, and from there we will move on to the different pathophysiological situations arising due to irregularities or due to certain disease states of this network. So, to start off with, if you recollect the last slide, to start here. So, we are into, so this is what we dealt, spatial and temporal summation. So, I told you, so there are neurons which are sitting at different places in the network. So, if I call this as, so just before that, put it like this. So, this is continued on week 6th, lecture 1. Okay. So, if this I consider as space 1, space 2, space 3, likewise, and you can keep on increasing it. So, they are sitting at different places in the space, yet they are connected, right. So, the communication channel is happening like this. Say for example, this X, or if you go with an or rather this E, the way we have, if this is excitatory. So, this one is sending a signal to this space 2, all these different 3 different neurons. Fine. So, now few things could happen. One is at what time who is receiving the signal. So, for example, the signal comes here. So, if this is the first one to receive a signal, then if this is the second one, and if this signal, or if suppose there is a connection, something like this or something like this or something like this, and if this is the third one. So, you see you can put certain time into it. So, for example, this one receives a signal at time t 1, this one receives a signal at time t 2, this is receiving a signal at time t 3. So, let us redraw it, that will make more sense. So, suppose this is one neuron space, this is another neuron in another space. Now, it is sending signal to this. So, this is neuron A, this is neuron B, this is neuron C, okay, in the network, and the signal transfer is always according to the direction of the arrow. Okay. Now, suppose, and let us assume this one is excitatory, this one is inhibitory, this one is excitatory. Now, suppose at time t 1, this A sends a signal. So, this is excitatory, this made this one to generate a signal, and this gets transmitted further down. But just imagine at time t 1 this generated a signal, at time t 2 this receives a signal, this the neuron C, but exactly at time t 2 imagine another signal which is inhibitory signal which arrives here exactly at time t2, this time t2, then what will happen? Then the signal which is, suppose I consider this as in blue as the positive signal or excitatory signal, in red I consider this as negative signal. So what will happen? There will be an addition of the quantity of the positive and the negative signal. So I haven't given it a quantity, I have just given it a sign, and the resultant will decide whether this neuron, which is the C neuron, which is an excitatory neuron, whether this will transmit the signal further or not, or what will be the amount of signal which will be transmitted. So, for example, I say this positive signal is 20 unit, okay, some 20, and the negative signal is say 5, then the total signal which will be transmitted will be 15. Or while this one is getting excited, with a slight delay, this inhibitory signal comes even before it generates an action potential, part of it. So there may be slight reduction in 5, this may be like becomes 4, so your resultant will become 16 likewise. So this is the most simplistic system where I am showing you this, but just imagine if this becomes a very complex network where you have inhibitory signal, excitatory signal, self signal. So there is something, say for example a neuron out here, it sends part of the signal and the part it falls back on itself. So if we have talked about synapse or the synaptic zones, synapse, there is something called OTAPs, or you can break it as autosynapse. So in other words, a part of the signal will be fallen back. Okay. Similarly, it could have part of the signal going to the next one. Okay. So there are millions of possibilities which could happen in a network, and what we consider as behavior, consciousness, these are emergent properties, as they call it, of the network. It is the summation of the network what makes us who we are, and it's a very, very complex network. And if we look at biology with respect to time, so you will observe that the last century is the century of cellular and molecular biology. There are a lot of molecular events, cellular events which have been defined. Earlier to that, it was more of a naturalist way of, so you see the gross structure and its behavior and all that kind of stuff. Okay. So what you used to see, so the evolution of biology is like that. Initially, we never had the tools. We were more concerned about the behavior, the overall behavior of the network. But the last century saw where we tried to understand the individual function or molecular events happening at the individual nodes of the network, which includes, say for example, what is happening in a single neuron, what is happening in a small network like this, or what is happening in a complex network like this, likewise. But now, the next generation of physiologists, neurophysiologists, neural engineers, the idea will be much more to understand the holistic viewpoint. Now we have a sufficiently good understanding about single neuron or couple of neurons out there, but then how these things really leads to the word what I use, the emergent behavior or emergent properties. That is something, emergent properties or emergent behavior that is something which will boggle the scientist in the centuries to come. So, it will be a long journey where we will be kind of trying to understand these emergent phenomena. But at this point, having said you this, I add few other interesting details about this network. So, we have already talked about that at the synapses, if this is the synaptic cleft where neurons are transmitting their signal through the neurotransmitters. So, these neurotransmitters can be either excitatory or inhibitory, you have talked about it. But what is important is that these synapses where the neurotransmitters are released, say for example, if this is the presynaptic terminal and this is the postsynaptic terminal, this hand, the one which I am waving now is the postsynaptic terminal and this is the presynaptic terminal, and they are like in close opposition and very close, they are not in contact but very close to each other. So, this one, the one which I am waving now, if this one secretes neurotransmitters and this one receives the neurotransmitter, the one now which I am waving, which is the post one. But this neurotransmitter has a finite residual time in that small cleft. If that residual time exceeds by some XYZ reason, do not worry about what is the reason at this time, then that will create a lot of trouble. Why it will create a lot of trouble? Just think over it, just visualize what will happen. So, this one secretes at time t1, this one receives at time t2, and immediately this one gets excited and the signal gets transmitted along this neuron. But if this prolongs, what will happen? If this is an excitatory one which is getting secreted, right. Say for example, just let me put it on the slides, that will make more sense. So, this is the one which is sending signal and this is the one which is receiving signal. So, this is A and this is B. Now, this one generates an action potential at say T1 and this one is supposed to receive at T2, and this is where the neurotransmitter are released and they bind here and they open up the sodium channels and they generate the necessary action potential, right, to travel. But so there is a small T2 minus T1, a small time window when this event is happening. Now, this time window is very critical. If this time window prolongs, say for example, this comes as delta t. If you have any delta t plus some other delta added to that, some other small time, then what will happen? If this one, if A is secreting your excitatory neurotransmitter, then this B will become hyper excitable, because it will have much more positively charged ions or sodium ions which will be getting through the voltage gated channels. Such situation could lead to a different level of activity of the network at times, and at times such situation could be pathological. How? I do not know how many of you have seen this. If you have seen a epilepsy patient, the symptoms are very interesting. Whenever a epilepsy patient gets a bout, epileptic bouts, what happen, I mean, the person start all of a sudden like lose contact of the system and kind of start shivering and fall down. And once they regain consciousness, if you ask that person, do you remember anything what happened, they will say no. They become dizzy and kind of and there is enormous movement of all the hands, legs and everything, like, something like this. What happened exactly is, from the neurophysiological perspective, in the vast network, say for example, if I consider something like this, if this is the network, I am not drawing the individual neurons now. Okay. If this is the network, I am just keeping a gap just to tell synaptic. At certain zones of the network, there is a hyper excitability. All of a sudden, for a transient period of time. So much so that the individual kind of faints out because it cannot compensate for that high metabolic activity, because whenever these cells are synthesizing such vast amount of neurotransmitters and secreting. So, obviously, for that process to happen in a fraction of a time, that requires enormous amount of energy to be produced by the cells, and the individual who suffers from such thing faints out. It is almost like, there is another analogy by which you can understand this. It is something like, imagine a four-way crossing like this. So, four-way crossing has a rule, right? So, if these ones, when these one will, they will go straight, or they go like this, or they go like this, fine. Similarly, when the other party goes from this side, they either will go like this, they will go like this, they will go like this. Similarly, if you are coming from the top, so something like this, and or you will go straight, and exactly by the same token, if you are coming from this side, you will go like this, or you go like this. Now, synapses has to be imagined something like a four-way crossing. Multiple synapses at one point, and one is talking to the second one is talking to the third one, and there is a lot of crosstalk happening without obstructing each other. It is a very interesting zone where these crosstalks are happening, and one has to understand that if in a four-way crossing, if the traffic light goes haywire, if the traffic lights are not working, there will be nothing but collision, like this, and just you can use that analogy to understand what will happen in epileptic bouts. In the case of epilepsy, there is an hyper excitability of the system, and the system goes totally haywire. But where this happens, which part of the brain is involved in it? So, as of now, I haven't talked to you about the demarcation line about epilepsy, or the demarcation line between the central nervous system problems, peripheral nervous system problems, and other accessory problems. So, let us do something. Let us draw a map out here for us to proceed further, which all aspects are we going to deal here. So, what we will do, we will talk about, briefly we will revisit the, in the light of this, briefly we will revisit a stretch reflex arc, which we have already started, but we have not added all the necessary components of the action potentials and everything. Second, out here as an extension, we will talk about neuromuscular junction. In short, sometimes they call it NMJ. Then we will talk about brain anatomy, and spinal cord anatomy, which partly I have already talked. There we will talk a little bit about hippocampus and related disorders, which one we have already started. We'll conclude once I show you the anatomical location: epilepsy, AD or Alzheimer's disease, Alzheimer's. Then we'll talk about substantia nigra and Parkinson. In the spinal cord, we will talk about motor neuron disorder, which will include amyotrophic lateral sclerosis, or in short they call it ALS. Then we will talk about spinal cord injury, and within spinal cord we will talk about glial disorders. Remember, I have introduced about the glial cells, glial myelination disorders, where there we'll talk about very briefly about multiple sclerosis. Okay, and in this whole process, talking about the brain anatomy, the most emergent property or evolving property is memory and its models. In the light of whatever we have understood about the process itself, and while talking about this multiple sclerosis, we'll devote a little bit time about myelination in CNS and PNS, which is peripheral nervous system, and there we will talk about oligodendrocytes. So this will be our map, what will be following for next few classes to conclude this part of the course. Okay. So I will close in here. In the next class, we will continue following this map. Thank you.